So this just means at some point long ago when researchers were working on this particular receptor, they discovered that nicotine would activate some of them, but not all of them. And then they found this other chemical, muscarine, that would activate the other ones, but not the same ones nicotine activated. And then we ended up with these two different groups. And this is how we distinguish between them. And I mean, there's lots of examples of receptors that are named after the chemical used during research to discover them, rather than what actually binds to them in the body. Ryanidine receptors, that's a good example. There's no ryanidine in the body. Right? That was something that was used experimentally to get those calcium channels to open, in the sarcoplasm in particular. And so that's what they ended up calling them, right? Ryanidine receptors. But what actually binds to those receptors in the body? Calcium, right? Calcium-induced calcium release. Yeah, don't write this down. This is old stuff. That's chapter 9. It's an example of how we often name receptors after some exogenous chemical that's used to explore them, rather than what's actually binding to them in the body. When ACH binds to nicotinic receptors, what mechanism of action is triggered? What does that mean?
You have one acetylcholine for one receptor, and you open it.
So the chemical binds to the receptor, a channel opens, and that's it. Done. That was the whole purpose, to open that channel. And you didn't have to go through a bunch of steps or reactions to get to that opening. In fact, the channel and the receptor are part of the same protein. Can't get any more efficient than that. Indirect. What does that mean?
ACH to the receptor, to the G protein, to the ACP, to the cyclic AMP, to AMP, opens channels.
They're looking to see if you're doing that off the top of your head.
No, definitely not. You were talking really fast, and I was like, okay. So shorthand.
So paraphrase that.
I don't know.
The chemical binds to receptor, and a bunch of steps have to occur before you get the whatever it is, the opening of a channel, or the activation of an enzyme, or secretion of a product, or whatever ends up getting triggered in that cell. It's a bunch of steps to get there. Let's say we are trying to open channels, and we use the indirect method, and that ACH binds to the muscarinic receptor, which is indirect. How many channels might open as a result of ACH binding to one receptor if it's an indirect mechanism of action? Thousands, millions, tons. That's the upside, that's the benefit. With very, very little chemical, with very little first messenger, you generate tons of second messengers, other chemicals inside the cell that end up producing the cellular response. So one second messenger pathway that's used a lot is cyclic AMP. And so that's very possible that ACH binds to the receptor, activates a G protein, switches on adenylate cyclase, an enzyme that starts cranking out tons of cyclic AMP. And then that cyclic AMP is going to do all sorts of things in the cell. It could activate a bunch of enzymes, which then phosphorylate proteins, maybe channels, causing them to open. But from that one neurotransmitter, you can get thousands of second messengers, like cyclic AMP. And after it activates enzymes, each one of those enzymes, called kinases, can phosphorylate multiple proteins. So if you have 5,000 phosphorylated kinases, and each one, if you have 5,000 activated kinases, and each one phosphorylates a protein, you get at least 5,000 proteins doing something in that cell. And if it's a channel, you just open 5,000 channels. But if it's an enzyme, and you switched on 5,000 enzymes, in turn, they can do whatever thing amplified by 10. Meaning, once you switch on the enzyme, it doesn't make one product, it can make dozens of product. But you have 5,000 of them working at the same time. We'll talk more about that next semester. But it's amplification is really what the huge benefit is. You get an amplified response. What's the downside to indirect? Takes time. What's the upside to direct? Fast. super fast. So, mechanism of action is simply what happens once the chemical binds to the receptor. That leads up to the response. Switching on of enzymes, secretion of a product, opening of channels. That's what mechanism of action refers to. Everything that follows binding to the receptor. And when it's direct, there's one thing that happens following the binding. Channel opens, done. Locations. Where do we use nicotinic receptors? Muscarinic.
Target cells. And some CNS.
So if I ask for 2 for each of these, you've already got 1. Some CNS neurons, that covers both, right? So you remember that, you got both of them taken care of, and then you just have to remember one other one for nicotinic. I would go with NMJ, you already know it. And then there is only one other one for muscarinic. Target cells of parasympathetic fibers. So we can connect that to chapter 12. So in the spinal cord, or in the brain, and I've only shown you in the spinal cord thus far, but as you'll see when we get to chapter 14, we also have autonomic 2 neuron chains extending from brainstem nuclei. Now, if the preganglionic fiber is coming from the brainstem, how is it reaching the postganglionic fiber, and in turn, the target cell? Let's do it this way. What are the 3 parts of the nervous system? Brain, spinal cord, and nerves. Brain and spinal cord represent the CNS, nerves, PNS, all right? So how do these axons, how do these fibers reach the target cell? By passing through nerves, right? Bundles of axons, bundles of fibers in the PNS are nerves. So the nerve is going to connect the brain, the brainstem in this case, to the target cell. What do you call a nerve that extends from the brainstem? You have 12 of them. Cranial nerves. What do you call a nerve that extends from the spinal cord? You have 31 pairs. Spinal nerves. All right, cranial nerves. They can be motor only, they can be sensory only, they can be mixed. And that refers to the fibers that are within them. So how about cranial nerve 1, olfactory? All of those nerve fibers within that nerve do one thing. Carry sensory signals regarding smell to the brain. Sensory. How about cranial nerve 3? What's its name? The name tells you if it's sensory, motor, or mixed. Oculomotor. It's motor. And it tells you where it goes. What does that nerve innervate? Eye. Yeah. Not the eyeball itself, but something that is attached to the eyeball and moves it, and those are extrinsic eye muscles. How many of them do you have? Extrinsic eye muscles. 6. 2 oblique and 4 rectus. How many of those are controlled by oculomotor? 4. Yeah. What are the other 2 cranial nerves that control the extrinsic eye muscles? Abducens. Trochlear. So 3, 4, and 6. What does trochlear innervate? Superior oblique. What is trochlear named after? The trochlea, that little loop of connective tissue on the medial surface of the orbit that the tendon of the superior oblique muscle passes through to make a 90-degree turn and attach to the top of the eye. Right? So the eye is here, the muscle belly is here, there's this little loop, and then the tendon goes through it, and then makes a turn and attaches to the eye. So we're naming the nerve after that little sleeve of, or sling, that little sling of connective tissue, the trochlear. So it innervates the superior oblique. How about abducens? What is that named after? What it does to the eye. Abducts. Well, which muscle abducts the eye? Turns it laterally. Lateral rectus. So what does abducens innervate? The lateral rectus muscle. All right, so back to this. We're looking at muscarinic receptors on the target cells of parasympathetic nervous system fibers, postganglionic fibers more specifically. And we're just seeing how these fibers reach the target cell, and that's by passing through nerves, right? All neurons that extend from the brain and spinal cord reach their targets by passing through nerves. You never see a neuron by itself, right? They're all bundled together into a nerve. And for that matter, you don't have an axon by itself in the CNS. They're bundled, and they're bundled based on function. What do you call a group of axons that carries the same type of information in the same place? A tract. How are tracts typically named? Like the corticospinal tract. So the first part of the name, where it originates. The second part of the name, what it innervates, right? Where it goes. The destination, origin and destination. So where does the corticospinal tract originate? Cortex, cerebral cortex. And where does it terminate? Spinal cord, corticospinal. It is a bundle of what nerve fibers? Pyramidal cells, also called upper motor neurons, also could be referred to as the first order neurons of the somatic motor pathway. How many neurons in that chain for the somatic motor pathway? 2. What, upper and low? Yeah. What does have a 3 neuron chain? Sensory pathways. To get from an exteroceptor in your skin all the way to the cerebral cortex, 3 neurons. First order, second order, third order. Which one's the first order? The one going to the cerebral cortex or the one forming the receptor in the skin? So for a sensory pathway, where would you start? At the receptor, right? That's the first order neuron. Well, what classification of neuron is that? You can give me functional class, you can give me structural class. Unipolar, structural class. Functional class, sensory. Yeah. So all of those sensory neurons in the peripheral nervous system, and this is aside from special senses, which are unique, so general senses, they're all unipolar. So every sensory neuron whose fiber is passing through a spinal nerve is a unipolar neuron. Where do you find the cell bodies of this unipolar neuron? Dorsal, median sulcus, ventral, median fissure, dorsal roots. I'll use this as an imaginary line, so this is the dorsal, or posterior side, and ventral, or anterior side. All right, so this is dorsal root, this is ventral root. So what's right next to the dorsal root? A ganglion. The dorsal root ganglion, the DRG. And what's in there? cell bodies of those unipolar neurons. So the unipolar neuron technically has one process that comes off of the cell body. But these neurons are often described as having two processes. In which case, we're not referring to what comes off the body, we're referring to the axon and the two parts of it. So here's the body, has a single axon, no true dendrites, but then the axon splits, right? And then this is the two processes that they're talking about. So if this is the receptive ending side, maybe a little Meissner's corpuscle there, what do you call this half of the axon? Or in other words, what do you call this process? Peripheral. And this one? Central. And why is peripheral process called peripheral process? Because it's fully contained in the peripheral nervous system. And central process is called central because it goes into the central nervous system. So receptors way out here, that peripheral process goes all the way to the dorsal root. The cell body is right there in the dorsal root ganglion. And then from that point on, the rest of the axon is considered the central process. It goes into the CNS. If this unipolar neuron is conveying sensory information from the lower limb, where is that information headed when it reaches the brainstem? The medulla, and in the medulla, a nucleus called nucleus gracilis. What if the information is coming from the upper limb? Nucleus cuneatus. So those are two nuclei in the medulla. So that first order neuron, that unipolar neuron whose central process entered the spinal cord, it then goes all the way up to the medulla before its first synapse, before it synapses with the second order neuron. So the cell body of the second order neuron is in nucleus gracilis. Right? We're still going with the foot, lower limb. The axon of the cell body that's in nucleus gracilis, it projects up. Right? This is a projection fiber. Where does it go? Where does that second order neuron go? Where does all sensory information go before reaching the cerebral cortex, with the exception of olfactory input? Thalamus. The thalamus, like nucleus gracilis, is gray matter. It's a collection of cell bodies. So the cell body of the third order neuron is in the thalamus. And then where does it project? Where does the axon from that third order neuron cell body go? Primary somatosensory cortex. Right? So it's just putting it into a pathway. So we're taking all these individual structures that we've identified between chapter 11, chapter 12, and mapping it out in a pathway, seeing how it all fits together. We can add more. Now that we've covered, just yesterday, part of the spinal cord section from chapter 12. We looked at the white matter that the axons pass through to go up to the medulla. So for sensory input coming from the lower limb that's headed up to nucleus gracilis, what structure do the axons, and I'm talking about the central processes, what white matter structure do they pass through or actually form to reach the nucleus gracilis? Fasciculus gracilis. Well, what's a fasciculus? A tract. Yeah. So this axon, or the central process of it, is bundled with others that carry similar information, going to a similar destination, and it forms a big bundle that's a tract, or fasciculus, and it's named after the nucleus that it goes to. So fasciculus gracilis takes signals to nucleus gracilis. Nucleus gracilis then sends signals up to the thalamus. Thalamus sends those signals to the cerebral cortex. We can add one more. Yeah, we should. We need to know. It's chapter 12 of the brain. what white matter structure that passes through all levels of the brainstem is going to contain the axons from the second order neuron. The axons whose cell bodies are in nucleus gracilis. What white matter structure is going to contain those axons that are going to head up to the thalamus? Medial lemniscus. There you go. So the axons of the first order neuron pass through fasciculus gracilis to get to nucleus gracilis in the medulla of the brainstem. And then the axons of your second order neuron pass through medial lemniscus to get to the thalamus. And then from the thalamus, those axons pass through what two white matter structures to get all the way to the primary somatosensory cortex. We're past the peduncles, we're all the way up at the thalamus, right? And the cerebral peduncle is right on the sides of the midbrain. So what band of white matter?
Reticulum?
Say again?
Reticulum?
What band of white matter is on either side of the diencephalon? Internal capsule. The internal capsule is on either side of the diencephalon. And the internal capsule actually separates the lentiform nucleus from the caudate nucleus. It goes right between them. All right, so then the internal capsule fans out. So maybe half of the axon that's going from thalamus to cerebral cortex is forming the internal capsule, and then after that it forms what? Corona or something. Corona radiata. So we just took a bunch of anatomy, a bunch of different structures, and connected them into a pathway. And that's what you eventually want to be able to do. That's where we're headed with all of this. Not just learning them in isolation, but how are they all connected? That was just a little tangent. I'll go back to where I started, yeah. But this is honestly how you should study. You just go, go, go. See how many things you can connect, see where it takes you. Right, so we were looking at ACH, muscarinic receptors, locations, where they're used. That includes parasympathetic nervous system target cells like cardiac muscle, smooth muscle, glands. So the anatomical nickname of the parasympathetic division is craniosacral. And that tells you where you find the cell bodies of your preganglionic neurons. So we'll do a different color for sympathetic. So what's this tapered, cone-shaped bottom tip of the spinal cord called?
Is that the
Cauda is all those
Oh, the conus.
nerve roots that come up. Conus medullaris. So you have one coccygeal spinal cord segment, and then above that, you have your sacral spinal cord segment. So the nickname for the parasympathetic division is craniosacral, because that's where you find the cell bodies of preganglionic parasympathetic neurons. What neurotransmitter is released from these preganglionic parasympathetic neurons? ACH. What receptor is it binding to on the dendrites and cell body of the postganglionic neuron? Nicotinic. Now, all of these cell bodies of all of these postganglionic neurons are clustered together into a ganglion, right? An autonomic ganglion. So that was one location where you find nicotinic receptors, at your autonomic ganglion. And it doesn't matter if it's parasympathetic, which these two are, or if it's sympathetic. Now, what was the anatomical nickname for the sympathetic division of the ANS? Functional nickname is fight or flight. Anatomical nickname reflects where you find the cell bodies of the preganglionic sympathetic neurons. Thoracolumbar. So what is that gray matter structure, this is from yesterday, in the spinal cord that you only find in the thoracic levels and upper lumbar that contains the cell bodies of your preganglionic sympathetic neurons? Lateral horn. In the lateral horn is a cluster of cell bodies, so a nucleus. What's the name of that nucleus found in the lateral horn? Visceral motor nucleus. What do we typically call visceral motor? Autonomic. And if you called it autonomic nucleus, that's fine. Right? That essentially means the same as visceral motor nucleus. But that visceral motor nucleus only contains cell bodies of sympathetic fibers, not parasympathetic. Right? So they're separate from each other. Preganglionic parasympathetic fibers originate in brainstem nuclei and sacral spinal cord segments from the cord. Hence, craniosacral division of the autonomic nervous system. Preganglionic sympathetic neurons, their cell bodies are only found in the lateral horns, which are only found in the thoracic and upper lumbar regions of the cord. Hence the name thoracolumbar. Craniosacral, thoracolumbar. Two nicknames remind you where you find preganglionic cell bodies. What neurotransmitters released from sympathetic preganglionic neurons? Same as the ones released from parasympathetic preganglionic, ACH. And again, it binds to nicotinic receptors on the postganglionic cell. The difference is, what neurotransmitter is released from parasympathetic postganglionic fibers? ACH. But how about sympathetic? Norepinephrine. So this is my shorthand way of saying acetylcholine binds to nicotinic receptors at autonomic ganglia. Doesn't matter if it's parasympathetic or sympathetic, right? So the green is sympathetic and the blue is parasympathetic. So again, neurotransmitter from parasympathetic postganglionic neurons, ACH. But what receptor is it binding to now? Muscarinic ACH receptors. And just for the shorthand, I'll leave off the ACH, just put muscarinic receptors. And then the neurotransmitter coming from sympathetic postganglionic. It's a biogenic amine, norepinephrine, and the receptors that it binds to. Adrenergic receptors. So going back to chapter 11, classes of neurotransmitter, you've got acetylcholine, it's its own class. You have biogenic amines. One of those that you need to know is norepinephrine. One place that it's used, secreted from postganglionic sympathetic fibers, and that's where it binds to adrenergic receptors on the target cell. What else did we need to know about ACH?
Sanitary or.
Right. So when it binds to nicotinic receptors,
Always the same.
and then muscarinic,
Sanitary or
what would it depend on then? It can't be the neurotransmitter because it's ACH, or the receptor because it's muscarinic, so it must be the cell. So you can have two different cells that both have muscarinic receptors, but when ACH binds to them, you can get very different effects.
You said, for nicotinic, acetylcholine receptors, you could call them nicotinic cholinergic receptors. Is there another name for the muscarinic
muscarinic cholinergic. In fact, the way it works is nicotinic receptors and muscarinic receptors are cholinergic receptors. So instead of saying nicotinic ACH receptor, you could say nicotinic cholinergic receptor, and then over here, muscarinic cholinergic receptor. In a similar way, and I was going to wait till chapter 14 to say it, but that's all right. These adrenergic receptors that norepinephrine binds to, there's two types. Just like there's two types of ACH receptors, nicotinic and muscarinic, adrenergic receptors come in two flavors as well, alpha and beta. So you have alpha adrenergic receptors and beta adrenergic receptors, which is just like saying muscarinic cholinergic receptors versus nicotinic cholinergic. Anything that has the word cholinergic tied to it means acetylcholine. Anything that has adrenergic tied to it is referring to norepinephrine or epinephrine, which we used to call noradrenaline and adrenaline. So the adrenaline rush that you feel